donc avec INBA. Thank you INBA for your collaboration. Donc le travail a été donc valorisé par 3 publications de renommée internationale, dont professeur Brahmé était vraiment très actif dans ce projet. Donc d'ailleurs les trois papiers ont été produits par professeur Brahmé. Maintenant, je passe au coproduit de l'olivier. Alors, donc, la suite après. Donc, alors, cette partie, en ce qui concerne l'olivier, donc c'est une partie de notre projet de recherche PRIMA, donc qui est chapeauté par professeur Gemrar Haddadi Hayat, dont moi, professeur Brahmi, docteur Jaoud et monsieur Watmani Toufiq sont membres. Alors, l'olivier est un arbre béni et millénaire. Il est valorisé, bien sûr, depuis longtemps par ses fruits et son huile. Il génère, par contre, des milliers de tonnes de coproduits. Alors, environ 4 à 5 tonnes de coproduits pour avoir 1 tonne d'huile. Alors, ces coproduits présentent des enjeux économiques et environnementaux qu'il faut donc saisir. Alors, je vais vous présenter maintenant une fiche de synthèse de ces coproduits en Algérie et je vais m'intéresser beaucoup plus à la quantité de coproduits générés. Alors, concernant les grignons, donc, qui représentent environ 35 à 40% du poids des olives, donc ils produisent environ 51 100 tonnes par année. Pour les margines ou les eaux résiduelles, nous avons environ 200 000 tonnes par année. Pour les feuilles, 5% qui représentent donc 5% du poids des olives. Par contre, pour les noyaux et les eaux de végétation, nous n'avons pas donc de chiffres qui concernent ces 2 coproduits. Alors, pour la valorisation et l'application de ces coproduits, donc il est basé sur la composition, bien sûr chimique et biochimique, sur leur composition chimique et biochimique. Pour les applications, donc, ils ont beaucoup d'applications, à savoir l'extraction des antioxydants qui peuvent être utilisés dans le domaine agroalimentaire ou bien pharmaceutique et cosmétique. Alors, je vais vous présenter un schéma simplifié donc, qui concerne la valorisation de ces coproduits. Par rapport aux grignons, donc, on peut produire des composts, des biogaz et de l'huile de grignon. Les margines, donc, ils sont riches en composés phénoliques, donc on peut extraire ces composés phénoliques et procéder à l'enrichissement des matières, de produits alimentaires. Donc, on peut les utiliser aussi pour le traitement biologique, c'est-à-dire pour les utiliser pour traiter les eaux usées, et on peut aussi produire des biogaz. Pour les feuilles, donc, ils sont riches en antioxydants naturels. Donc, on peut les utiliser pour la fabrication des aliments fonctionnels et dans le domaine cosmétique. Alors, pour les noyaux, on peut les utiliser donc, pour produire des briquettes de charbon qui peuvent facilement être utilisées pour le chauffage. Et on peut aussi produire de la biomasse. Alors, pour l'eau de végétation, on peut récupérer les composés actifs qui sont contenus donc, dans ce coproduit. Alors, c'est dans le cadre toujours du projet PRIMA de professeur Gemrar. Alors, nous avons entamé le travail, je pense, il y a 2 ans de ça. Alors, nous avons, Madame Brahmi, professeur Brahmi a encadré un mémoire de master. Elle vient de soumettre une publication donc, pour valoriser ce travail. Donc, le travail a consisté en la valorisation des feuilles de l'olivier et par enrichissement donc, du fromage frais. Madame Gemrar, ou bien professeur Gemrar, a travaillé sur le grignon d'olive. Donc, elle a extrait les composés phénoliques. Elles ont procédé à l'enrichissement du fromage frais toujours. Alors, pour les perspectives, c'est-à-dire le travail que je compte moi-même réaliser, c'est l'optimisation de l'extraction des composés bioactifs par des procédés innovants en utilisant un coproduit naturel comme solvant d'extraction. Et bien sûr, je vais essayer donc de préparer des emballages à base de ces extraits, à base des extraits de ces différents coproduits. Alors sur ce se termine mon intervention. Je vous remercie pour votre attention.
Thank you very much, Professor Boulkbesh, for this interesting presentation. Next, we will hear from Professor Matheus Preciusidid Inmaculada from University of Complutense of Madrid, Spain. Professor Rashad will give us her biography.
Donc, professeur Immaculada, donc de l'Université de Madrid, dans le département de nutrition et sciences des aliments. Donc, elle est spécialisée beaucoup plus en chimie alimentaire, valorisation des sous-produits agroalimentaires, prébiotiques et technologies de traitement qui visant à améliorer la fonctionnalité et les ingrédients issus de ces produits.
Presentation is about from waste to work, reducing food waste in the valorization chain. You can begin your presentation, Enma.
Thank you very much. Good morning. First of all, je désolé, I don't speak French, and I'm a bad English speaker also, so sorry. If you lose during my presentation, don't worry, it's not your fault, sure it's my fault. But I offer to reply any question that you have in the breaks in the Congress to have a nice talk if you want. Then, firstly, I would like to thank to the University of Bellaya to be here. It's a pleasure for me to be in Algeria with my colleagues. So thank you very much, especially for the organizing committee of this interesting Congress, for my invitation to be a part of the scientific committee and also giving the talk today. My presentation is going to focus in a proposal to try to find a way for adequate and efficient way to valorize food waste. I'm going to divide this talk in 4 parts. Firstly, I will introduce the idea of food sustainability. Then I will try to discuss about which is an agro-food byproduct. Then in the part 3, I'm going to try to propose you the idea of a valorization chain that from the point of view of my research group is the way to do an efficient valorization of byproduct. And finally, I'm going to give you some final remarks as conclusions. So, starting with food sustainability. 1/3 of the food produced worldwide never gets consumed. So, this is a huge amount of discarded food. Therefore, reducing food loss and waste is essential. Indeed, United Nations General Assembly in 2015 proposed us the 17 Sustainable Development Goals. I'm going to focus in the goal 12 that propose to ensure a sustainable consumption and production. And in its target 3, propose the preventing food loss and reducing waste generation. So, which is the difference between food loss and food waste? We know that residues are generated along the entire food chain. But if we move to the first stages, such as harvesting, processing, distribution, the discarded part, the leftover, should be considered food losses. However, if we move to the final stages, such as supermarket, food services, as hotels or restaurants, and at home as final consumers, the discarded parts should be considered food waste. And these discarded parts are very difficult to recover and to reintroduce in the food production chain. I start mention the Sustainable Development Goals. So maybe it's important to define which is sustainability. Sustainability is the management of resources responsibly so that the future generations can benefit also of them. It brings together environmental care and social well-being, of course, with an economic balance to get both. To incorporate sustainability to the agro-food area, we need to establish a sustainable food system that can be defined as one that provides food security and nutrition for everyone, while preserving the economic, social, and environmental basis for future generations. This definition is found in the Farm to Fork strategy included in the European Green Deal, a deal from European Union to comply the Sustainable Development Goals from United Nations. This strategy is creating a healthy food environment and empowering their consumers mainly through the knowledge of food labeling. But the most important thing that proposes this strategy is the fight against food waste and food losses through research and innovation and promoting, of course, a global transition, not just in the European Union. As I mentioned, the residues are generated during the whole food chain. But if I focus in the first stages, mainly in primary production and processing, we have the 30% of the residues generated of the total of the residues in the food chain. These residues, as I mentioned, are easier to reintroduce in the food chain. So we should consider them byproducts. So what is a byproduct then? The definition of a byproduct could be parts of food generated as a result of a production process in which their production is not the main purpose, and therefore they are usually discarded during the different stages of that process. But if I stop here, is the same definition for byproduct or for a waste or residue? So which is the difference between waste and byproduct? Waste is thought to thrown away, to be eliminated. However, byproduct is thought to have an use. Is going to be used later. Moreover, can be used directly. It should be an integral part of a production process, and the subsequent use have to meet all relevant requirements such as any other ingredient. Here we have some examples about byproducts or wastes generated in the food industry. I'm going to focus in the first one, the fruit and vegetables industry. We have here that the discarded parts are peelings, stems, seeds, cells, bran, and more. If the industry thrown away these parts, incinerating or landfilling, they are treated as waste. But if we try to get an use for them, we are considering them byproducts. And maybe we can notice that all these discarded parts retain many interesting, valuable compounds, including bioactive compounds such as polyphenols, carotenoids, and of course nutrients such as proteins or dietary fiber. So I'm going to move to the third part of this talk. I want to propose to you how my research group think about to valorize this byproduct. Is the proposal of follow a valorization chain. My research group have several research lines, but our main research line is the identification and characterization of byproduct to obtain added value ingredients. We have studied several byproducts, but now we are focused more in apple byproduct, apple pomace, legume pods, and also artichoke byproducts. The valorization chain is the proposal to valorize agri-food byproducts efficiently in adequate way. That means to increase the value or at least to restore the value that has been lost. For that, from my research group, we propose 5 steps. First, a selection of the byproduct, then a treatment to apply depending on the byproduct selected, followed by evaluation of the effect created for the treatment applied, and then finally studies of quality and bioactivity to finally get a potential functional food product. I'm going to start with the first step, the byproduct selection. We have to focus in food production. We need to know the byproduct yield, and also we have to know the retention of bioactive and valuable compounds. In this table, we can see the amount generated of some agri-food byproducts worldwide. I'm going to focus in the last one, in the apple pomace, apple byproduct. We know that apple is one of the most important fruit crop worldwide, but in Europe is the most important one. During the processing of apple, it's generating between 25 and 30% of the in residues of the total of the product. So we have here a huge amount of byproduct. And also we know that these byproducts retain interesting valuable compounds such as dietary fiber or polyphenol. I would like to show you some of our articles related with byproduct selection. In this case, we have the identification of legume byproducts such as pea pod, broad bean pod, or okara, that is a byproduct from the soybean beverage production. We know that they are potential source of functional compounds because we study their composition. And of course, they are very rich in dietary fiber, more than 50% of the composition. In this other example about apple byproduct, we study the composition of the dietary fiber because we know that it's very rich in this compound. And we see that it retains a lot of pectins, a type of dietary fiber. We move to the second step, the treatment. Now we have selected the byproduct. So which is the more suitable treatment to apply to this byproduct? We can divide the treatment in 3 types: bioprocessing, extraction, the conventional ones, and the assisted extraction. I'm just going to focus in the assisted extraction based in sustainable technique, such as supercritical fluids, microwaves, ultrasounds, and hydrostatic pressure, and other treatments, such as electric pulses or irradiation. Regarding the bioprocessing, as Professor Lila before tell us, is the use of microorganisms to obtain valuable compounds, and the most known bioprocessing is fermentation. But also, inside of this kind of treatment, we can use specific enzymes. And we have several publications regarding this topic. I'm going to focus in this one about the use of an enzyme, specific enzyme, cellulase, food-grade cellulase, to release oligosaccharide and to solubilize dietary fiber. If we move to the extractions, regarding the conventional ones, I want to show you one of our patents. In this case, a world patent about the obtaining of a fat binder from biomass resulting from beer production. This fat binder is based in chitin and chitosan polymers, and we obtain it using alkali hydrolysis as conventional extraction. Moving to the assisted extraction, I just want to focus in hydrostatic pressure. That is one of the main techniques that my research group use, and maybe it's not very known, such as ultrasound or microwave. Hydrostatic pressure processing is equivalent to a cold pasteurization, and it consists, we put inside of a plastic bag the byproduct that we want to treat. Then we introduce it in a vessel, and we fill the vessel with a liquid able to transmit pressure, such as water. Then we submit the byproduct to a pressure ranging 1,000 and 6,000 bars, a very high pressure, more than the deepest zone in our planet, that is the Mariana Trench. Then we empty the vessel and we obtain the hydrostatic pressure treated sample. We have here some examples about this, the application of this treatment, hydrostatic pressure, here to improve the functionality of okara, the byproduct from the soybean beverage production. We saw here that the higher treatment intensity, in this case 4,000 bars, was the treatment to increase the solubility of dietary fiber. And what is important to solubilize dietary fiber? Because soluble dietary fiber is related with the able to ferment for our colonic microbiota. So if we increase the solubilization of dietary fiber, we are increasing the value of the byproduct to obtaining a potential prebiotic ingredient. Another example that we use is the combination of hydrostatic pressure assisted by food-grade enzymes. This is a novel approach for getting a synergistic effect. What that is mean? We obtain the solubilization of dietary fiber, like with the hydrostatic pressure alone, but also we reduce the molecular weight and release oligosaccharides, so we can increase the prebiotic effect. Another example, in this case, in apple byproduct, we can hear that when apply hydrostatic pressure, we solubilize the pectins. So the use of the high pressure improves this solubility. And when we use hydrostatic pressure assisted by a food-grade cellulase, we solubilize the fiber and decrease the molecular weight of these polysaccharides, as in the case of okara that I mentioned before. Well, in the last type of treatments, I just want to mention the irradiation, specifically the UVC irradiation. This treatment is used to control the ripening and also to control the contamination, but also can retain or increase bioactive and nutrients. Indeed, in European Union is a strategy to obtain novel food by increasing vitamin D content, such as in bread, milk, or mushroom. In my research group, we use this irradiation in several byproducts, but I would like to show you 2 articles that I have, my research group have done in collaboration with my colleagues here in Bellaya. The treatment of UVC light of an Algerian underutilized date fruit, the Phoenix dactylifera, and also the residues from the date syrup production of this date. And what is the main result? We have here that we can increase the polyphenols profile of the underutilized date and also the residues after the date production, the syrup production, sorry. The third step on the valorization chain is the evaluation. So we need to know if which is the effect of the treatment applied. Monitor the treatment. So we can use several techniques for this purpose, but I just want to tell you about the chromatographic technique, mainly molecular exclusion, that serve us to monitor the solubilization of polysaccharides and oligosaccharides, and also a structural analysis such as nuclear magnetic resonance or the example in the slide, the scanning electron microscopy, that allow us to see the cell wall disruption. And we need to see this disruption to explain the solubility of the polysaccharides from the cell wall. The fourth step is the quality. As we know, quality is a complex term, so usually it's divided in types. We have hygienic quality, that is the absence of microorganisms in the treated byproduct. Also the nutritional quality, to study the composition of the treated byproduct, especially to quantify the amount of the bioactive or the valuable compounds. Also, we have the technological quality to study the physicochemical properties, to understand how to formulate the byproduct well in a final food product, and the sensory quality about the study of the modification of sensorial attributes. Just two examples about our investigation about technological quality. In this case, we study the technofunctional properties that are enhanced with hydrostatic pressure. For example, the hydration properties, such as swilling capacity, but also the oil holding capacity or emulsifying capacity. So it allows us to formulate better the byproduct treated than the byproduct untreated byproduct. An example of sensory quality, we have here an evaluation of the orange pulp byproduct treated with pasteurization to see a slight modification of this treatment in the treated byproduct. For the last step, we have bioactivity studies, and we can do different bioactivity studies: in vitro assays, batch cultures of cellular lines studies, or in vivo assays. Regarding the first one, I am here an example to show you about monitoring the antioxidant capabilities of the treated byproducts, or in this case, in which we compare the different antioxidant capability that we measure with the identified polyphenols, to know a relation between if a polyphenol increase, we can get an increase in of the antioxidant capacities. Regarding batch culture and cellular lanes, just two examples. One in batch culture about the effect of oligosaccharide from chitosan over the human fecal microbiota, in this case to look for the prebiotic effect, or in keratinocyte cellular lines, in this case, looking for the protective effect of triterpene-rich extract from apple byproduct against reactive oxygen species. And in the last case, in vivo assays, we did in the past studied with animals, because now we are more focused in the first studies, the bioactivity studies in vitro and cellular lines. But in animals, we can see the modulation of gut microbiota, for example, in rat feta high-fat diet enriched with okara, or in this case, that we study the potential hypolipidemic effect of the diet enriched in dietary fibra from artichoke byproduct in Syrian hamster. So now, just some final remarks for finalizing my talk. Food sustainability aims for a sustainable food system that minimizes waste generation. The consideration of agri-food byproducts for those waste discarded, at least in the early stages of the food chain, can help achieve food sustainability. Agri-food byproducts are sources of bioactive compounds, which opens the door to their utilization and specifically to their valorization. And the valorization chain that my group proposed as the correct valorization represents a systematic strategy for an efficient valorization through the selection of suitable byproducts, the design of a sustainable process, the monitoring of the process to see the effect, and the comprehensive assessment of the quality and bioactive of the final product. Thank you very much for your attention. Shukran.
Thank you very much, Professor, for this very interesting presentation. Thank you for the insightful point of view into valorizing different byproducts from food industries. For now, we invite all the participants to make a pause and have lunch. We will resume after half an hour. Thank you.
Donc, s'il vous plaît, c'est juste, on va continuer les prochaines conférences après le déjeuner. Donc, l'assistance, donc les participants sont invités au hall pour manger. Et bon appétit.